In this activity, you will investigate if there is life in a mysterious substance. But as an opening, I suggest that you start by discussing microorganisms. What are microorganisms? Where do they live, etc. And if you have your student's manual, you will find some pictures of different microorganisms there. And then you may tell them that you, by the help of some researchers, have found a very mysterious substance. You can make up yourself where it has been found. Here in Norway, we usually say it's been found far up north on an island called Spitsbergen, where it's very, very cold. And actually, researchers from NASA go to practice how to behave on a new planet. So, well, here we have this substance. And we wonder if there's some alive organism in there. Is there a microorganism in here? How, in so case, can we wake it up and make it multiply and really become alive? And of course, there is no really strange organism here. Actually, this is a mixture of baker's yeast, some flour, and some sand. But it looks kind of mysterious, and if you have a microscope or a magnifying glass, it's nice to take a look, and especially the dry yeast will look a kind of mysterious and small cylinders, very regular, and they get more curious. Before you move on, then, you have to discuss what's special about life. How can we say that something is alive? And one thing is what life on Earth is made of. Actually, all life on Earth is made of the same molecules. You have DNA, you have protein, you have fat, you have carbohydrates, and all living organisms need water. In addition, life multiplies, and one more thing you can discuss is that all living organisms need energy to survive and to build their bodies and to build next generation, etc. And this energy can be different kinds of molecules. For example, for us humans, we use fat, protein, and carbohydrates, sugar as energy sources. But for other organisms, they can also be different kinds of molecules. But one thing is in common for all organisms when they produce energy. You usually start with rather big molecules, and they are broken down to small molecules that are set free again as gases. So, for example, I breathe out now carbon dioxide all the time, and that's a result from the metabolism going on in my body. So one way to trace life is actually to see if gas is developed. And usually you will have to help your students start thinking this way, but start it as an open question and lead them on the way. And then you can show them the equipment you have ready. You have either bottles or flasks like I have here, and you can then set up different combination with the mysterious substance in these flasks, and you just put a balloon on top, and if a gas is developed, it will be filled by gas. And as you see here, yesterday I did that with this mysterious substance, and I added, because I think it needs water, and I thought also it needed sugar, so I added sugar and water, and you can see it woke up. It became alive, and it started to metabolize the sugar and produce gas. So there is certainly something live in here. But this is the final solution. With your students, you can investigate different combination. For example, one combination as a negative control should just be substance and balloon. And other very interesting combination is to try just oil, food cooking oil, and substance, because then they will really experience that without water there is no life. And from there on, you can move on to, this is brown sugar. You can combine sugar and oil, sugar and water, or only water. And maybe you also have other ideas that you can find something around you, surrounding you, that you want to test. And we suggest that your students work in group to put up a whole series of different combinations. But the details and how you organize it, it's really up to you. Maybe you want to be very controlling and structure the whole investigation, or maybe you want to let your students go more freely about it. And usually after 1 to 2 hours, you can see the result by measuring how much gas has been produced in the different balloons, and then you can conclude. So it's an idea to not start this late in the day, but start it before lunch, so you have time to conclude. Good luck.